La malaria è una malattia potenzialmente mortale, prevenibile e curabile. Interessa 89 paesi in tutto il mondo, dei quali i più gravemente colpiti si trovano nell'Africa subsahariana. Nel corso degli ultimi 15 anni, la lotta alla malaria a livello mondiale ha visto una riduzione della mortalità di quasi il 50%. Armati soprattutto di insetticidi, zanzariere, farmaci e diagnostica, si stima che finora circa 7 milioni di vite siano state salvate. Nel 2018 si sono riscontrati 228 milioni di casi e più di 400.000 decessi, dei quali il 67% erano bambini al di sotto dei 5 anni.
Il 17 novembre del 1970 l'Organizzazione Mondiale della Sanità dichiara ufficialmente che l'Italia è un paese libero dalla malaria. 50 anni dopo ci troviamo qui all'Istituto Superiore di Sanità per celebrare questo importante traguardo della nostra sanità pubblica e ricordare inoltre il ruolo cruciale che ha avuto l'Istituto nel raggiungimento di questo traguardo. L'istituto viene inaugurato il 21 aprile del 1934 col nome di Istituto di Sanità Pubblica, assorbendo in pieno attrezzature e personale della stazione sperimentale per la lotta antimalarica, che aveva sede al Palazzetto della Farnesina a Roma. Diviene quindi il centro di riferimento nazionale e internazionale per gli studi sulla malaria e per le attività di lotta antimalarica.
Ci troviamo nella sede dell'Istituto Superiore di Sanità di Via Giano della Bella, nell'aula dedicata ad Alberto Missiroli, medico, insigne malariologo, che fu il primo direttore del laboratorio di malariologia, rimasto in carica dal 1934 fino al 1951, anno della sua scomparsa. Ha rappresentato sicuramente la figura chiave della lotta alla malaria in Italia. Portò avanti l'attività di ricerca, di formazione e di aggiornamento del laboratorio anche durante il periodo bellico, nonostante le difficoltà del momento. Grazie a finanziamenti internazionali e alla sua profonda esperienza di malariologo, nel 1946 pianificò prima e poi organizzò e diresse il piano quinquennale per il risanamento dell'Italia, che prevedeva l'eradicazione della malaria. L'Agropontino ha rappresentato sicuramente uno degli scenari più significativi della lotta alla malaria in Italia, anche perché nel 1943 l'armata tedesca in ritirata, per ostacolare l'avanzata degli alleati, aveva distrutto numerosi impianti idrici presenti nell'area, ricreando di fatto l'ambiente paludoso idoneo allo sviluppo delle Anopheles e vanificando così la faticosa opera di bonifica messa in atto su tutto il territorio nazionale alla fine degli anni '20. Negli anni successivi l'Agropontino fu nuovamente colpito da epidemie di malaria di notevoli proporzioni. Nel 1945 le tabelle sanitarie dell'epoca riportano oltre 180.000 pazienti malarici assistiti.
Ci troviamo a Latina, al Museo della Terra Pontina, in compagnia della direttrice del museo, la dottoressa Emanuele Francisconi. Grazie innanzitutto per la disponibilità.
Il museo ha appunto una sezione relativa alla malaria con i reperti dell'ex istituto antimalarico, dove abbiamo ricostruito infatti il laboratorio e l'ambulatorio e tutta quella che è stata il percorso di igiene, di profilassi che a partire dagli anni '20 addirittura con quello che è stato l'istituto antimalarico e poi il comitato antimalarico che chiude appunto negli anni '70, abbiamo ripercorso quello che è stata questo grande lavoro di eradicazione della malattia. Come dicevo, i reperti sono tutti originali e sono dell'ex comitato provinciale di Littoria, il comitato provinciale antimalarico di Littoria e poi di Latina, che opera dal 1920 sino poi agli anni '70, in cui, terminato il suo compito, l'istituto antimalarico chiude i battenti. Qui abbiamo ricostruito parte del laboratorio con tutti i reperti originali che sono stati, come dicevo, una donazione della ASL e che facevano parte del laboratorio dell'ex comitato antimalarico.
Dagli anni 70 in poi, in Italia le attività di controllo della malaria autoctona sono state sostituite da quelle legate alla sorveglianza della malaria di importazione e allo studio dell'anofelismo residuo, come ad esempio quello realizzato nell'area della Maremma Toscana in provincia di Grosseto. Negli ultimi 10 anni in Italia si sono registrati in media circa 700 casi di malaria importata l'anno, a fronte di una media europea di oltre 7.000 casi. L'Istituto Superiore di Sanità, quindi, in stretta collaborazione con il Ministero della Salute, assicura un sistema di sorveglianza basato sulla conferma della diagnosi microscopica e l'aggiornamento regolare della situazione epidemiologica dei casi importati e l'attivazione del sistema di allerta che prevede l'indagine epidemiologica immediata in caso di sospetti casi autoctoni. L'impegno dei ricercatori dell'Istituto Superiore di Sanità nella lotta contro la malaria viene portato avanti attraverso un'ampia attività di ricerca e tramite il coordinamento e la partecipazione a numerosi progetti nazionali e internazionali, come quelli con la cooperazione italiana in Africa. Nonostante gli innegabili progressi raggiunti fino ad oggi, la malaria rimane una delle malattie infettive più diffuse al mondo, a causa della resistenza del plasmodio ai farmaci antimalarici e delle zanzare anopheles agli insetticidi e alla mancanza di un vaccino efficace. Ostacoli che rendono sempre più difficile realizzare i programmi di controllo realmente efficaci, soprattutto in Africa.
Con l'impegno e la collaborazione di tutti sarà possibile raggiungere il traguardo più importante, un mondo senza malaria.
We can start. Good afternoon, everybody. First, I would like to thank all the speakers for their participation in this webinar of Plasmodium vivax, and all those who are following us connecting and streaming. Let me introduce you Dr. Luigi Bertinato, presenting an official welcome from the presidency of Istituto Superiore di Sanità. I give the floor to him. Please, Dr. Bertinato.
Thank you, Carlo. Good afternoon, ladies and gentlemen. I officially welcome all of you to the conference on Plasmodium vivax malaria. And the official welcome is on behalf of Professor Silvio Brusaferro, which is the president of the Italian National Institute of Public Health. Today, it's a great opportunity to celebrate the Malaria-Free Italy 50th anniversary. I think it's really an event for all Italian colleagues. 50 years ago, malaria was free from Italy. WHO declared Italy free from malaria. I think we have to go back to 1918 when Professor Alberto Missiroli set up the School of Malariology. And thanks to the School of Malariology of Professor Missiroli, the institute I represent today was set up in 1934, devoted to malaria at the beginning. And Professor Missiroli was the key player, the scientist who played the major role in the fight at the beginning in setting up the first national plan against malaria in 1946. I think that the history of this institute is part of malaria. Malaria was the idea of setting up an institute, and now the institute is devoted to all public health threats, not only malaria. Today, we have also the opportunity in this first meeting to celebrate the last historical scientist that in the Institute was devoted to malaria prevention and control, Professor Giancarlo Maiori, which unfortunately passed away last July 6. he was the director of the Laboratory on Parasitology. He was a director of the WHO Collaborating Center on Tropical Neglected Disease. He was also member on the technical review panel of the Global Fund to fight AIDS, TB, and malaria. I think he was really the ambassador of the malaria culture across the globe that we have in Italy. That's why it's really a pleasure, an honor to officially open the conference. Thanking all our speakers for their contribution, but also thanking Carlo Severini and his staff to having put a lot of efforts in creating the two events on malaria to celebrate the malaria-free Italy. And it's with this wish, I wish all of you a successful meeting, and hoping that very soon malaria will be an event that can be part of the history. A lot of research was done recently, and I think today is an opportunity to focus on Plasmodium vivax. Thank you, Carlo. I pass you through the responsibility to manage and organize the event. Thank you, and have a good meeting.
Thank you very much. Thank you, Luigi. So we can go ahead with the program. Just two brief considerations from myself. Can I have the slide, please? Okay. Okay, just a brief consideration, because we have to respect strictly the webinar timeline. 2 points. The first point is referred to this graph that you can see in this slide. Although this graph refers to the past situation in Thailand, where, as you know, falciparum and vivax coexist, in my opinion, it represents anyway a good example to show you. As you can see, every time that an effective drugs or drug combination is used against falciparum, and then the number of cases of falciparum decreases, vivax, on the contrary, remains constant, and therefore the proportion approaches or reaches 50% each. Next, please. So the message, in my opinion, is clear and direct. Plasmodium vivax cannot be controlled, defeated by implementing the same control activities that are implemented for falciparum. It's clear that it deserves specific funding, specific project, specific therapeutic protocol, and so specific research and control activities. So this message has been received by many researchers starting in the early 2000s. And like you, all of you who are here today to present your talks, let me say that I'm proud to be a part of this scientific community. The next, please. So the second point is concerning Europe. The message is that we can speculate that in case of emergence of malaria in Europe, probably Plasmodium vivax will be the responsible Plasmodium species. We have to have in mind this fact. And actually, the next, please. The Plasmodium vivax actually hit hardly in the past, even at higher latitudes of Europe. This is how the vivax patients or those who were retrospectively identified as vivax patients was described by the English doctor John McCulloch. You see that the description is very, very sad, and describe the very unhealthy people. John McCallum is considered the precursor of the discipline of malariology. This is the British physician. So the next, please. In summary, the Congress directly focuses the attention of the revised traditional concept of vivax as benign parasites, and recognize the importance of an update of vivax current research that often is neglected. So that this meeting is an opportunity to consider vivax infections as a priority to overcome the challenges for malaria control. Okay, that's all. Next slide. Okay. Thank you for your attention, of course. And the next slide. So this is thus you remember the program. For those who are connected in streaming, you can say the email for question at this dedicated email address. During the presentation. The selected question will be presented at the end of the presentation. So at this point, I think we can go ahead with the program with the first speaker. This is George Snunu from Department of Infectious Disease Models for Innovative Therapy, CIA University Paris-Saclay. I think in a nice place near Paris. Fontenay-aux-Roses in France. George, please. Thank you for your participation.
Thank you very much, Carlo, and I'm honored and to have been invited to this conference. And I'm kind of a bit nervous about talking about Italian malariology when I'm not an Italian myself, but I can claim that I'm a malariologist. I have added this little word, some contributions of Italian malariologists, because malariology was born in Italy, and the Italian contribution is so huge that to summarize it in 30 minutes is impossible. I would therefore not summarize it, but I will just pick up a few points, which I hope will be of use for people to think about malaria. So, next slide, please. Right, so you probably all know that malaria is a very old disease, not so much that it was there with us before, is that we knew that what malaria was from the antiquity until now. I mean, from the antiquity, people knew what malaria was, what its symptoms were. The major problem was that nobody knew how malaria was acquired. And since it was associated with swamps, the idea came that it was due to the bad air, hence the word malaria, and that it was somehow transmitted by these kind of emanations. In order to study any disease, we would need to know where the pathogen is and what it is in order to study it. And all the knowledge that people had acquired from antiquity until the late 1800s pointed to the air, and that remained what was thought. With the advent of the germ theory of Pasteur and Koch, people started to look for parasites and for the pathogens of malaria, and it became very important, in particular because Europe, which suffered from malaria a lot, and in particular Italy, the European nations were expanding into the colonies, and they really needed to know how to deal with malaria, which was a very important and major disease. Now, I've put here this business about ignorance and knowledge. Now, usually people say the reverse, that it's knowledge is valuable and ignorance is dangerous. Well, I think in science it's the reverse. Why do I say this? Because of dogmas. Dogmas are what drives science to a certain extent, but dogmas can be good and dogmas can be bad. And usually with a little bit of knowledge, one can have a dogma which is wrong, and with a lot of ignorance, one can imagine dogmas that will turn out to be right. So it is pardon. Excuse me, some things I can't switch off on the computer. Anyway, I'm going to start with I think, the original sin and mistake of the Italian malariologist, and I think that I could only identify one. And next slide, please. The first scientific publication to say that they've discovered the malaria parasite was that of Tomasi Crudelli and Edwin Klebs, both of whom were very eminent microbiologists in the late 19th century. And they've isolated from the water of the Pontine marshes a bacillus, which they called Bacillus malariae. They injected the broth of this bacillus into rabbits, which then became feverish, and they had an enlarged spleen, which is what you see on the right-hand side. And they concluded that this must be the parasite that causes malaria. Now, at least Tomasi Crudelli still believed that for the next 10 to 15 years. Given that he was a prominent malariologist and that he was a senator, he became a senator in 1892, a lot of people it slowed down the research in a lot of people, but not all of them. Because next slide, please. This is basically, this is the early landmarks in malariology. This is which I'll be discussing in the next few slides. As you know, next slide, please. The actual discovery of the parasite was made by Laveran in 1880, just 1 year later after Tomasi Crudelli published his work. And he had observed these parasites into the blood of infected soldiers in Constantine in Algeria. He got the Nobel Prize for his discovery, as it was considered as one of the most major discoveries at the time. Despite the fact that he studied malaria for the next 20 years, for the next 40 years, he died in the early 1920s. He always believed as a dogma that the parasites all malaria parasites were of the same species and that just changed from one form to another. It was the Italian malariologist who, next slide, please. Next slide. It was the Italian malariologist, indeed, who actually named the parasite species and confirmed by observations on patients infected with malaria, of which there were plenty in Italy, that there were actually 3 types of parasites, one of which was highly dangerous, which were the Plasmodium falciparum. So it was a tertian parasite, but it was malignant. One of them, which was quartan, which was given the name of Plasmodium malariae. And the last one, a tertian relatively benign or relatively less malign malignant parasite, which was named, next slide, please. Next slide, in 1890 by Grossi and Felletti. Now, Grossi and Felletti, in the name vivax only appeared briefly in one short publication of Grossi and Felletti in 1890. And to just show, to kind of illustrate how difficult this was, at the time, remember, the microscopists were looking at live blood, unstained, unfixed, and with the best imagination, this is the kind of picture of malaria parasites that they had. So one has to say that the observation powers of the early malariologists were phenomenal. And indeed, having discovered that there were 3 parasite species, despite the fact that Laveran always counted this, a lot of studies were being conducted now that they knew what the parasite was, to try to see how the parasite is transferred from man to man. This was still quite a mystery in the 1890s, and many researches were done over the next few years, none of which kind of really pointed it out. There was still the idea that maybe the parasite floated on water and was picked up by just imbibing the water or inhaling particles containing the parasite. Next slide, please. The solution to the problem came from the intellectual thinking of Manson, who had demonstrated that filaria can be transmitted by mosquitoes, and who by the late 1890s, was quite old and was living in London. And he convinced a relatively young major, Ronald Ross, who was sent to India in the Indian Medical Service to follow the parasite from the mosquito, from the human, into wherever it goes. And he considered that it probably will go into the mosquitoes and therefore Ronald Ross, intellectually directed by Patrick Manson, kept looking at parasites, at mosquitoes that have fed on hosts that are infected with malaria. The parasites that were easiest to study were the malaria parasites of birds, because, as you know, birds have a profusion of malaria parasites, and these were certainly much easier to study in India, funnily enough, because the Indian medical services didn't really much appreciate Ronald Ross working on malaria and humans. And therefore, the first description came in 1897, the first breakthrough, when he saw these funnily pigmented bodies on the guts of the mosquitoes. Now, in my opinion, unbeknownst to Battista Grassi, Battista Grassi was doing similar work with the mosquitoes, because there was this idea that mosquitoes transmit malaria, which is also an Italian idea coming back all the way back from the days of Lancisi in the 1717. And he demonstrated in humans that it was indeed mosquitoes that transmit parasites, and he actually completed the cycle himself and, of course, some of his colleagues. And he further demonstrated that the parasite was transmitted only by anophelines. Now, just to let you, Ronald Ross was absolutely incensed and considered that Grassi was a pirate and a thief and a plagiarist. And it started a Roman dispute, which lasted until the death of both Ronald Ross and Battista Grassi, which I think was a great shame, because it deprived malariology of a collaboration that should have made malaria move much faster. But Ronald Ross was not a very pleasant character, I must admit. This is my personal opinion. And he certainly was quite ignorant of zoology. I mean, it's you just have to look at the quality of the drawing that the two of them have to show that the difference, what it was. To compound matters, it was Ross who received the Nobel Prize, and Battista Grassi was excluded from this. But that's another talk. Now that the malariologists have now discovered what is the parasite and they know how it is transmitted, they have discovered the sporozoite, which is made in the salivary glands. And the problem was, how does the sporozoite go? Where does the sporozoite go when it goes into the body? The view started to appear amongst Italian malariologists that there must have been a small part of the cycle of this life cycle which doesn't take place inside the blood. But next slide, please. These views more or less stopped, and these experiments to explore them stopped, the second that Fritz Schaudin, another eminent protozoologist, described in one article that he saw from the blood of a patient infected with vivax, he saw a sporozoite going inside a red blood cell, and here's the picture that he's drawn to show that. Now, this was the only observation that was made, and I must admit, for the next 20 years, 30 years, nobody, although they've tried to reproduce this observation, but it was such a convincing idea that the sporozoites go straight into red blood cells that actually none of the research of malaria kind of took place to study the fate of the sporozoite during this time. Therefore, one can say that a single observation can precipitate a dogma which will last for 20 years. During these 20 years, during this time and after the observations of Fritz Schaudin, practically all malaria research was conducted on observations that were made in the field and on observations made in patients that were recruited and observed. In some cases, treatment was withheld for a few days to observe the course of infection. And although people learned a lot about malaria, it was not possible to experiment on malaria in this time. And it is indeed the ability to experiment on malaria which has advanced substantially our knowledge of malaria and started to put into question the views of Schaudin. Next slide, please. Experimental malaria was due to a single man. The single man is Julius Wagner-Jauregg. He was a neurologist in Vienna who, after long researches and long studies and long trials, came upon the idea that it was fever that can cure tertiary syphilis. Now, people who get tertiary syphilis end up insane, and once it declares, then they die within 1 or 2 years. And it was a plague in Europe with a lot of tertiary syphilitics, nearly filling up 60 to 70% of all mental hospitals. Now, in 1917, he had the opportunity to take a few, as you can see on the small picture on the right-hand side, a few mLs from a patient with malaria and injected it into a paralytic who was suffering from dementia paralytica, and he allowed that fever to proceed without treatment for as long as the patient could bear. And he demonstrated in his first study published in 1917 that actually 7 out of 9 patients were practically, I wouldn't say cured, but improved immensely, considering that the disease beforehand was invariably degenerative and fatal. This was a major advance. Over the next years, malaria therapy, became to be known, malaria therapy, became widespread throughout the world. This is from Argentina to Russia to China, and in particular, it was in the United States and in Europe. Julius Wagner-Jauregg got the Nobel Prize, I think, in 1927 for his, or 28 for this observation. Now, in a lot of the malaria therapy centers, malariologists became associated in order to help provide the material to infect people. Because if you want to infect a lot of people, especially if you want to infect them by mosquito bites, then you really need a malariologist who understands how the parasite works and how to kind of conduct mosquito breedings and feeding, etc. Throughout the time, the malariologists took major advantage of the fact that they had now a large number of patients who were being infected experimentally with malaria to conduct a series of what I would call observational experiments. They did not devise the experiments and recruit people to infect them, but given that the fact that the people were going to be infected, they could decide how and when best and what with to infect them. And over the next 30 or 40 years, so until the advent of penicillin, malaria therapy has fueled acquisition of knowledge on the clinical aspects of malaria, its transmission, its immunology, how the parasites behave. And I have put the names of those who are most associated with malaria therapy and the knowledge from it. And quite honestly, anybody who wishes to call himself a malariologist who hasn't read some of their work, if not all of their work, is going to have difficulties convincing me that they understand malaria properly. Because the observations are fascinating and sadly very poorly known. Next slide, please. It is indeed one of the first observations made by the Liverpool School people, headed by Warrington York, that actually started making people think that this business about the sporozoite going directly into red blood cell was actually not quite correct. And to summarize that experiment on the left-hand side here, if a person is infected by sporozoite by mosquito bites, by inoculation of sporozoites, and that person is treated on day 0 and 2 after inoculation with quinine, at the time the only drug available, then that person had nonetheless a primary infection and then a relapse. The relapse was important in these cases because relapses were not cured by quinine. And that was a major problem because although quinine was helpful for falciparum, as Carlo was saying, you can't approach vivax the same way you approach falciparum. And it was well known from the beginning that quinine does not cure relapses. On the other hand, if the person was infected with sporozoite inoculation and then treated with quinine 7 to 8 days after the mosquito bit, then the primary infection did not occur. Now, this made it clear and proved that there was some sort of phase, call it about 7-8 days, during which the sporozoite is refractory to quinine. And the thought was that maybe the sporozoite takes 7 days to develop into the red blood cells, was the kind of leading theory of this. Now, that theory was disproved, in a sense, by experiments conducted in the mid-1930s when people were inoculated, again, that's on the left-hand side, people were inoculated by mosquito bites, again, whether falciparum or vivax. And what the experimenters did then is take a large amount of blood from the person within a few minutes, half an hour, 3 hours, 6 hours a day, up till day every day for the next few days and that blood was inoculated into a naive volunteer. And what these observations showed, and they were done by Boyd in the early 30s, by Suka first, and then by Hamilton Fairley, an Australian with the Australian Army in the early 1940s. What they demonstrated is that if you take blood immediately after a mosquito bites, immediately, that's within a few minutes, then you can infect another person with that blood. But if you stay more than an hour, then for the next 5-6 days for falciparum and 9 days approximately for vivax, the person who receives the blood stays uninfected. And only after 9 days and after 6 days for vivax and falciparum respectively do people become positive, showing that actually it wasn't sporozoite in red blood cells that were just lying there, that it must be that the sporozoite went to a place in the body which is not accessible to the circulation. In other words, it was an exoerythrocytic cycle or an exoerythrocytic phase that must have occurred there. Now, by the time these experiments were done, the idea that exoerythrocytic phase, which was in a sense rejected earlier, became fashionable. And the first person who showed an exoerythrocytic phase, next slide please, was Giulio Raffaele in 1936, at the time working with bird malaria, because bird malaria were used quite extensively in the first 30 years of the last century, simply because they were easier to handle than humans and to experiment with. He described for the first time parasites living inside non-erythroid cells in birds, and that was followed up by work confirmed by James and Tate in England, and the race was on to find out in humans which is the organ or the site in which the pre-erythrocytic parasites are, or the exoerythrocytic parasites are. This discovery was made in 1948 by, next slide please, by Short and Garnham, initially working on Plasmodium cynomolgi in 1948, and very quickly after they demonstrated parasites inside the liver and no other organ of the, after sporozoite inoculation, of macaques infected with Plasmodium cynomolgi, they did the same work with humans infected with Plasmodium vivax experimentally, and following that a few years later with falciparum, and following a few years later with ovale, and then with P. malaria. Nearly in all these cases, it was a volunteer who was inoculated with hundreds of thousands, if not millions, of sporozoites, and then a piece of their liver, a little biopsy was taken, and then they managed to demonstrate this. So by 1948, it was a big year. So this is, you remember, this is 58 years after discovering the parasite, and more than that, and 48 years after discovering the transmission, 50 years after discovering the transmission of the parasite, in order for the cycle to be now considered as completed. Given that this is what was that it was in the liver, now the idea of the relapse became, they come back to the problem of the relapse. They've solved the problem of what happens to the sporozoite the first couple of days or week. And then the question is, where does the relapse come from, came to the fore. And the view from that of these observations, next slide please, was that there was an exoerythrocytic cycle, and although already in 1948 they had observed on 100-day post sporozoite inoculation some forms in the liver that were parasitic. So the idea was that once the sporozoite is inoculated by the mosquitoes, it goes and infects hepatocytes, and the hepatocytes will then, once the parasite goes in the hepatocyte, the schizons will release merozoites. Most of these merozoites will go out into circulation, but the idea was that a few of them will actually reinvade another liver cell, and that liver cell will keep that cycle in the liver going until such a time that the merozoites coming out from these liver cells that have been infected a second time, hence the word secondary, will then precipitate, for reasons that we don't know why they come out, precipitate another clinical attack, which would be known as the relapse. Now, this idea stayed as a dogma, in a sense, or as the hypothesis considered as correct for the next 30 years. It was through the driving force of Wojciech Krotowski, who was convinced that, wasn't too sure that this is really what was happening here, that Garnham and Krotowski and many of the colleagues, London School, with the help of the American CDC, went and redid some of the experiments in cynomolgi. Next slide, please. And demonstrated, and here's the slide, the original slide on the left-hand side, where you see this kind of white dot on the top panel, is when they observed the parasite into the liver of a monkey, and they considered that this was not so much a form that comes from a secondary cycle, exoerythrocytic cycle, but it was a dormant form. And at the suggestion of Miles Marcus, they called it the hypnozoite. And the studies that kind of went after this, now the dogma has changed from the secondary exoerythrocytic cycle to the hypnozoite hypothesis or dogma, in which what the idea is that when a mosquito injects, and this is the current dogma today, is that when the mosquito injects sporozoites of Plasmodium vivax, that most of these sporozoites are of a type that will grow fast, develop, and create and give the first primary infections, but that some of the sporozoites are destined to sleep and to remain dormant in the liver and only activate at a later date. We still don't know what precipitates that activation for them to grow again, to give this a secondary relapse, not only one relapse. In some cases, you can have up to 10 to 20 relapses occurring over the next 1 or 2 years in this. So this is basically the state of affairs that we have with respect to the relapses and what causes them today. And of course, today, the problem of the relapse is still major because primaquine is the only drug that we have to deal with that, to which we've added now tafenoquine, a drug of the same class. And both of these drugs are unfortunately not suitable to give on a mass drug administration level because they are dangerous for people who have glucose 6-phosphate dehydrogenase deficiency. And therefore, the race is still on to try to find a drug which is effective and that can be given to a lot of as a mass administration. So next slide, please. The question remains, have we solved the problem of relapses, or is it still a persistent enigma? To my view, it is still an enigma. We still don't understand how the relapse pattern changes geographically. We still don't understand why parasites activate or relapse at certain times and not others. And for some of the strains, this is rather regular. And we still don't know, actually, whether relapses are only due to a dormant hepatic parasite. Now, over the years, people have had different theories. I'll start with the one in the middle, that vivax was observed once in the lymph nodes. Nobody did these studies. This is 1939. A patient was inoculated with P. vivax sporozoites, and a day later, biopsies were taken from the limbs. And as you can see, this is a picture of the sporozoite found in the lymph. It looks like a very healthy sporozoite to me. But of course, there was no proof that that sporozoite was infectious. And none of this study has not been, similar studies have not been done since. Recently, Robert Ménard and Roger Amino showed that you can have parasites going into the lymph. They can go in and infect cells other than the liver, but it was not demonstrated that they were infectious or indeed that they are not infectious at all. So there is still a question mark about what happens to the sporozoite after inoculation, whether they all end up in the liver and infect the liver cell. On the other hand, Irène Landau described in the 1990s in rodent malaria extracellular parasitic forms that look like a bunch of merozoites bunched together inside a little cocoon inside the spleen and of animals infected with the parasite. And again, and she called them merophores. There was no demonstration or proof that these are infectious, but nonetheless, this is not explored. Now, I go back to the idea that the spleen is an important, we all know that the spleen is important in malaria, but one person in particular, and I think forgotten, is Maurizio Ascoli, who in the mid-1930s, and I think 1938, has published to say that he considers that the spleen is actually a reservoir for red blood cells. And there's some evidence that this is quite correct. It is a reservoir of red blood cells, and it's probably a site where red blood cells are manufactured and made, apart from the bone marrow. He also considered that the parasite, because the red blood cells are kept in reservoir into the spleen, and they're kept in reservoir because of the way the spleen functions and its circulation is that there must be also a reservoir for the spleen. So he devised this treatment, which is called the Ascoli treatment, in which he associated the injection of quinine to treat people with the injection of adrenaline, because adrenaline contracted the spleen, and therefore, in his words, it would expel the parasites and make them available to quinine. And this was, I think, accepted in Italy and used quite a bit as a treatment, the Ascoli treatment. But it has not kind of been accepted very broadly. And I know that nowadays, by observations done in Papua New Guinea, there's been very strong proof that the spleen actually harbors the majority of vivax parasites, at least during chronic malaria. That it is actually in the spleen that you might have a cryptic cycle whereby the spleen is full of parasites and the peripheral blood is empty. Therefore, please keep your imagination active and don't believe all the dogmas that you read or all the observations that can become fact. They are factual, but for which the hypothesis is only a hypothesis. And therefore, we still haven't fully solved all the problems of the vivax and of the relapse. And it'll be quite interesting to know how this will evolve and how the role of the spleen, which is still considered as only a place where parasites get killed, and now is probably going to be considered as a place where the parasites live. Next slide, please.
Sorry, George, you have 2 more minutes.
I'll finish in 2 more minutes. Here are a few of the dogmas that are still, I think, a bit wobbly. Some of them are falling down. Clinical severity is fallen down, that the parasite is not, vivax is benign. Duffy negatives can't be infected is now shown to be not right. And as I say, sequestration is in the process of being drawn. There will be probably other dogmas on the transmission and how vivax transmits and how malaria is acquired that will probably be broken. Next slide, please. Now, I wish to put some of the known of the prominent Italian malariologists, and I'm sure I've omitted many, and there were many to omit, and I haven't put any of the people who are still alive today in order not to upset anybody. But it is the work of these great malariologists and probably the first 30 or 40 years of the 20th century that laid the foundation of malaria eradication. As I say, was mentioned, Misiroli was one of the first to use DDT and clear malaria from the areas of Frosinone area in which he studied. And of course, the bonification of the Pontine marshes. And the amount of publications that they have is, I wouldn't say phenomenal, but is very large and very substantial. But it is very sad, next slide please, that the vast majority of their work is hidden in journals that exist in Italy and that don't exist anywhere else. And I've noted here some of the few of the journals which are very difficult to find, but in which most of these publications are. And I would like to point out that the first journal devoted specifically to malaria, I've highlighted it in pink, was first published in 1899, and it generated then the Bollettino Malariologica and then the Rivista di Malariologia, both of which have an incredible number of very interesting papers, the vast majority in Italian. And I would like to leave a suggestion to the direction of the ISS is to please try to make these available electronically to people who are interested, because the treasures of observations and of ideas on malaria that are present in these journals is phenomenal. I will leave you with this idea, and thank you for your attention.
So
I'm
thank you.
happy to take questions, of course, and criticism.
The question, I think, at the end of the presentation.
No problem.
Like other question to get. Thank you very much, George, for this interesting presentation and for your tribute to the Italian malariologist. Thank you very much. We can go ahead with the next speaker, Professor Stéphane Picot from Institute of Parasitology and Medical Mycology at University Hospital in Lyon. The presentation is Vivax, Malaria, Drug Resistance, the Inevitable Loss. Please, Stéphane.
Hello, everybody. Thank you, Carlo. I'm very thankful to you for your kind invitation. I'm very proud to be part of this nice panel. And I will give a short outlook of drug resistance during vivax malaria. Next slide, please. Drug resistance with vivax malaria is not really a surprise. We can go back to 1949, where Seaton and Lurie were able to induce resistance to proguanil by injecting the same strain to different patients during 20 months. 10 years later, same kind of experiments were conducted to induce pyrimethamine resistance in patients. And finally, pulmoquine resistance was also induced in vivax malaria using this kind of model. So next slide. It's not really surprising to see in 1989 the first 2 cases of therapeutic failure with chloroquine in 2 soldiers contaminated in Papua New Guinea. Next slide. And this phenomenon has increased during the last 3 decades, and we can summarize the problem with 3 points. The first point is the capability of Plasmodium vivax to invade reticulocyte, and this is responsible for the lack of culture and the difficulty to obtain in vitro tests to examine the sensitivity of this parasite to the different drug. The second point is that it's difficult to get a radical cure because of the hypnozoite. So there is pulmoquine, probably tefenoquine, but it's not that easy to get a radical cure to treat these patients. And the third point is the capability of Plasmodium vivax to produce early gametocyte in the first cycle of the growth of the parasite, and this is involved in the difficulty to get an elimination of this disease. Next slide, please. Today, chloroquine is first-line treatment in many countries. Not very clear, but it's many countries, and the recommendation for systematic use of pulmoquine is unfortunately not that efficient in the field. ACTs are also promoted to be the first-line treatment in Asia, also with pulmoquine, and there's a question mark about the role in the future of tafenoquine. Next, please. If we are going down to the use of the first-line treatment, you can see that in America or in Eastern Mediterranean region, the chloroquine is still the first-line treatment with no treatment failure above 10% approximately. But the situation is quite different in other places. If we are going to Africa, to West Africa, the transmission of vivax malaria is not that high, but in Mauritania, for example, the chloroquine was still effective in 2012. We have not much results since then. But if we are going to the east of Africa, the sensitivity is decreasing, and many countries have switched to other drugs, in particular artemether-lumefantrine for Somalia, Sudan, and Ethiopia, and Madagascar is using artesunate amodiaquine with very good efficacy. Going to the southeast and western Pacific region is completely different. You can see that in Indonesia, the sensitivity of chloroquine has disappeared years ago, and now Indonesia is using DHA piperacin as a first-line treatment. Cambodia is has moved again to artesunate mefloquine with a very good efficacy today, but there is some stress about the sensitivity of mefloquine. Artemethyl-mefentrine is still the first line in Timor, in Laos, in Malaysia, but there is also very bad signal from Papua New Guinea, Solomon Island, Vanuatu, with an increased failure rate with artemethyl-mefentrine, which is a very important problem. The situation could be different in Vietnam or in Philippines. So next slide. So it's not very easy to define what is Plasmodium vivax drug resistance. It is starting with a patient, of course, but the patient has an immunity, has an age, has some comorbidities that could have an impact on the drug sensitivity. Self-treatment or delay before treatment may also have an impact on the efficacy of the drug. This patient has been infected in a specific area with entomological index, relapse pattern, either tropical or temperate, and a specific first-line drug policy, and all these points have also an impact on the drug sensitivity. This patient has been infected by a Plasmodium vivax strain with previous exposition to drug, with a wild type or mutant type of specific genes, and a specific rate of hypnozoid formation. And again, this has an impact on the drug sensitivity or resistance. And finally, this patient has been treated with a drug associated or not with primaquine. Was it a good regimen? What was the dose adherence for primaquine mainly? And we know that in some countries there is a circulation of about 30% of fake drug. Donc all these points are important to measure the drug sensitivity with drug resistance during vivax malaria. We have a patient with symptoms, with fever, and we need to measure the parasite clearance time. We need to have drug dosage in whole blood, and we need to have a parasite genotyping. So measuring drug resistance is not that easy. Next slide. We can consider that Plasmodium vivax multiplication rate is approximately 74 per 48 hours. So the delay per parasite clearance over 48 hours may lead to a 4-time increase in risk of recurrence. And we can see that the parasite clearance half-time is expected to be 4.5 hours with a specific dose of chloroquine. Next slide. But there are many confounding factors to be not related to the drug to be taken into account. The entomological, the patient, the regional, the concomitant use of primaquine, but also the weight adjusted dose. Chloroquine is frequently underdosed in children below 5 years. It should be 30 milligrams per kilogram instead of 25. And next slide, the main confounding factor. Next, please. The main confounding factor is related to the fact that vivax relapse cannot be distinguished genetically from recurrence or new infection because of the well-known frequency of different genotype in the liver. Next slide. Years ago, some of us were attending a meeting organized by WHO just to prepare the method for clinical trial on antimalarial drug efficacy, and there was a recommendation. I like that recommendation, but the recommendation is no recommendation was given for genotyping Plasmodium vivax because of the interpretation of genotyping in the context of relapsing infection. Next. At that time, we were looking for molecular marker with Carlo, and we described the Plasmodium vivax MDR1 gene with a specific point mutation at different codon. This was associated later with therapeutic failure, but controversial results have been obtained by different authors. And next slide. Rick Price just obtained at that time data demonstrating that EC50 of chloroquine was increased in patients presenting PVMDR1 mutation at position 1976. Next. But the link between therapeutic failure in Plasmodium vivax and mutation in this codon was not definitively established. And a recent paper from the team from Cambodia demonstrated that there is an inverse relationship. It could be an inverse relationship between the number mutation and the IC52 mefloquine, not chloroquine, but to mefloquine. And this also needs to be clarified. Next slide, please. Same kind of research have been conducted using a CRT marker for the same that in Plasmodium falciparum with different mutation. Next slide, please. And next, please. And these results were also not fully confirmed by in vivo test. A recent genetic cross have been conducted by Tom Willems with Plasmodium vivax sensitive and a resistant strain, demonstrating that the selection of a region in chromosome 1 with the chloroquine resistant transporter, the CRT, is located. But again, this needs more clarification. Next. Didier Ménard and his team in Cambodia have demonstrated that the Plasmodium vivax 12 proper domain could be linked to the autologous in Plasmodium falciparum. They detected some point mutation at different codon, but you can see that the incidence of this mutation is very low, and again the relationship between this mutation and therapeutic failure is not yet established. Next, please. So by the end of the day, we are in a situation where clinical trials are very complex to implement. In vitro tests are not available, and the molecular markers are not completely associated with drug resistance. So what we can do? I think that the first step is to move forward without chloroquine. And the first, the point is that chloroquine is still circulating in many countries where falciparum is also transmitted, and there is a risk for using chloroquine to treat falciparum wrongly diagnosed as vivax. And next, to come to an end, what we can imagine is to implement a kind of turnover between several ACTs as first-line treatment associated with a systematic chromocrine treatment after the G6PD deficiency have been excluded. And this requires the implementation of anti-malarial stewardship program with the same pattern that against falciparum, just to monitor correctly the drug resistance of Plasmodium vivax to different drugs. Thank you very much.
Okay, thank you, Stefan. Definitely, drug resistance is a very complicated task in vivax. Okay, thank you. We can go to the next speaker, Didier Ménard. Didier Ménard is from Malaria Genetic and Resistant Unit, Department of Parasite and Insect Vectors, Institut Pasteur in Paris, and talk about Plasmodium vivax transmission in DAFI negative people. This is another the dogma that is all overtake. Okay, so the problem, a technical problem with the connection with Didier Ménard. That we try to go with the next speaker, Marcus Lacerda. Marcus Lacerda is in line. Yes.
Sono qua.
Buongiorno, Marcus.
Buongiorno.
Thank you for coming. Okay, Marcus Lacerda for Tropical Medicines Foundation, Dr. Heitor Vieira Durado in Manaus, Brazil. So will present clinical severe forms due to the Plasmodium vivax infection and the current therapeutic approach. Please, Marcus.
Thank you very much. That's a very kind invitation. Usually people keep calling me to speak about severe case of vivax, which obviously reflect 20 years living in the field and working in a reference institution which sees all the complicated cases which are referred to this institution. Next one. So as you can see here, this is not an updated map from the MAP project in Oxford, but you clearly see that in the Brazilian Amazon, you have a huge prevalence of vivax, and that's probably what's happening with the Americas in general. Falciparum is being controlled over time. In Brazil, in these days, we report 90% of cases of vivax. So the whole country became a vivax endemic area, and that also reflects in many other countries in the Amazon. Next one. Well, I also have some old books and texts like George, and I still remember the first time I read the book from Marchiafava and Mignani, and things are very clear, and these people already knew from the autopsies they used it to perform in falciparum patients in Italy, and you clearly see here their first draw of hemozoin and how hemozoin was present in the vessels of patients dying because of falciparum. Obviously, they also saw a case of vivax, especially people during the winter going to the Italian Alps. But for some reason, nobody died, or they were not very curious about vivax patients. So in their reports, all we have is related to falciparum. So that probably refers to the perspective that vivax was considered to be a more benign disease. But as George has already described, there are many descriptions of severe vivax, and usually they were reported all together with other diseases. So if you take a look at the old literature, you're going to see that the severity was related to co-infections. So it's not novelty that you have a vivax patient being co-infected with some other disease and that leading to more severe disease. Next one. We waited until people in Southeast Asia described their first cases of severity in vivax to come out of the closet and show to the world that we also had that same experience, and that led to this paper published in the BMC Medicine, in which we used the same protocol, performing PCRs in all the patients in Manaus, which is A in your left, and in Bikaner in India, where they also report most of the cases due to Plasmodium vivax. And as you can see here, for over a year of patients being hospitalized in both institutions, which actually share the same infrastructure, you see here that the fatality amongst the admitted cases was not that much negligible. It was something around from 0.9 to 1.5%, which actually is a very similar fatality rate of falciparum in Brazil and in India. So if you take a look at those patients coming to you, you're going to see that, I mean, people actually die with the diagnosis of vivax malaria, and what's happening here. Next one. So as you can see, there are many ways for the development of severity. You see lung edema, which is a common clinical complication. You have people with cholecystitis developing jaundice. You have severe anemia, which is the most frequent type of complication. You have retinal commitment, you have acute renal failure. So these complications, they're pretty much well described in many places. Of course, different protocols have been applied worldwide, but they reflect to some extent that the type of severity of vivax is very similar to falciparum, except that you don't see in the peripheral blood the same amount of schizonts that you see in falciparum. Next one. So anemia is probably the most common type of complication, and the more you look for anemia, the more you see the impact of severe anemia, which can be as high as in falciparum. Next one. And that led us to try to understand what happens in the bone marrow. Next one. This is a recent publication from our group in which we saw the bone marrow of patients diagnosed with vivax on day 0 before treatment and on day 42. And what you clearly see here when you analyze the microscopy, you clearly see a level of this erythropoiesis which recovers at day 42, as you see here in the slide number C. And you also find parasites in the bone marrow, but not at day 42 anymore. Next one. And doing deep sequencing in these samples. Next one. And when you perform deep sequencing in these samples, what you see is that you have immune-related genes being transcribed differently, and that recovers by day 42. So usually at day 42, that's when you probably have a more regular bone marrow, and we are still exploring those immune-related genes which are changed, and that might explain to some extent part of the anemia which is seen, which is not necessarily because of destruction of red blood cells in the peripheral blood. Next one. Well, you don't have many autopsies in vivax patients. Here is a brief history of all those autopsies. As you can see, the first one being reported in 1895, and the patient who died because of streptococcal sepsis. That was the conclusion. Then you have patients with co-infections with falciparum. Billings then reports a patient developing hemoglobinuria. And then obviously most of these autopsies, they come from Bruch, who has described the autopsies in patients with syphilis already described by George. Next one. This is a picture that George has already showed to you. So Julius Wagner dealing with the COVID-19 of his time, which was syphilis, used malaria as the chloroquine of our times, and many people were treated with vivax malaria, and most of the histopathological knowledge that we have in these patients come from this time. Next one. This is then part of the autopsy series that we have reported here in Manaus, inspired by those cases which were reported with syphilis. This is a spleen in which we had hematoma in the surface, and that rupture led to bleeding. So it's not negligible the amount of patients who developed enlarged spleen and rupture of the capsule. Next one. Patients who have developed intravascular disseminated intravascular coagulation and died because of bleeding in their central nervous system. Next one. Most of the patients had lung edema, and when you take a close look in these slides, you see that they have neutrophils in the interalveolar space. Not many parasites are found here. Next one. In some cases, it's possible to detect. Next one. In some cases, it's possible to the previous. Yeah, this is a green parasite stain with anti-Vir for for vivax, so there are not so many. So I do not personally believe that cytoadhesion could be a major mechanism of death in these patients. Next one. And that we have shown in recent papers of our group in collaboration with other groups in Brazil, that you have some cytoadhesion of infected erythrocytes with vivax, and the more mature the forms, the more they cytoadhere. We have absolutely no idea on how is that cytoadhesion to organs like the spleen. Next one. That's probably the few comments I would like to address on the clinical complications, and in the next minute and a half that I have, I will show to you that there are some major challenges regarding the treatment of malaria. Stefan has already shown in detail some of this data. In Brazil, as he pointed out, we have no more than 10% of chloroquine failure, and it's not throughout the whole country. But if you look at thousands and thousands of cases over time, you're going to see that the recurrences until day 42, which probably represent better the chloroquine failure, you're going to see that those under 3 years of age are the most affected population, reaching sometimes 7.5%, which is totally different from other ages in which you have less than 5% of recurrence. Well, that might be a low recurrence rate, but that is enough to dismantle the elimination programs that we are envisioning in the recent times. Next one. This is a brand new data showing the impact of compliance, Primaquine compliance. So you have supervised Primaquine for 7 days. That's what we have been using in Brazil for more than 25 years. And you clearly see that when these patients, they are consented to be supervised, and you compare those patients with those who were not consented, but we got their data from the online system in the Ministry of Health, you clearly see that there is a difference in terms of the recurrences until day 180. So the major problem that we have been prescribing Primaquine to kill hypnozoites, but the fact is that even with 7 days, the compliance, the adhesion to treatment is not perfect. So if we don't have further tools to overcome that issue, we should not target elimination. Next one. And finally, the last slide is just to call your attention for the fact that Primaquine also in 6-PD deficient population, in an average of 85 cases per 100,000 Primaquine users, they could develop severe hemolysis when they are deficient. Nowhere this deficiency is being tested before treatment. In Brazil, we have reviewed 9 years of hospitalizations in our institution, and that's how we came to those numbers, which are pretty much numbers that reflect locally the impact of that use without the proper genetic testing. And in the next one, you're going to see a drug which has been developed over the last decades. You see here in the bottom, patients using chloroquine by itself, and in the top, you see in green and in blue the two groups, one who has used Primaquine for 14 days and the other one who has used tafenoquine for only 1 day. So these curves, they overlap, showing that the single dose tafenoquine in people who were tested previously for the 6-PD deficiency, you have virtually the same efficacy as compared to Primaquine 14 days. If that is the case with another ACT, another schizocidal treatment, we have no idea, but at least when you use chloroquine, tafenoquine replaces very well with the advantage of being used as a single dose. So that's a new tool. In Brazil, we are about to start the first implementation study in 2 municipalities in order to see the daily life of the use of tefenoquine and the G6PD deficiency test before that use. And next one, finally, once again, grazie mille.
Thank you very much, Marcus. Thank you. Very impressed with your presentation. So we have recovered the connection with Didier Menal, I think. I'm sorry, Didier, for the inconvenience. We are ready to for your presentation. So Didier Menal is the Malaria Genetic Resistance Unit, Department of Parasite and Insect Vectors at the Instituto Pasteur in Paris. The presentation is about Plasmodium vivax transmission in Duffy negative people. Please, Didier. Thank you.
Thank you, Carlo. You can hear me?
Yes.
You can hear me? Ah, okay. So it's okay? I'm sorry, I use a bad link for the connection. So, as you said, I will talk about P-vivax transmission in Duffy negative people, which is quite new, and we have some evidence over the 2 last decades that vivax is able to infect Duffy negative people. Next slide. Next, yes. So the dogma is that P-vivax interaction with the Duffy antigen require to invade human erythrocyte. Next slide. So this dogma was based on was based on several evidence. The first one was the discovery of the Duffy blood group in the 50s. The scientists observed the first one, which was the Phi A, and after 1 year later, they described the Phi B blood group, which is co-dominantly expressed, but they also observed that some individuals do not express the Duffy antigen and carry no antigen on their red blood cell. Few years later, the Bray showed that the distribution of the Duffy negative phenotype was highly prevalent in African population. Next slide. So by using epidemiological data, we observe that there is no overlapping of the distribution between the Duffy negative phenotype and the prevalence of P-vivax, especially in Africa. Next slide. The second evidence came from malaria therapy. So we already talked about that. And we observed that African Americans and Africans was highly resistant to P-vivax infection from different strains from different geographic origin, and with those which normally make an infection in Caucasian. And they also observed that the same ethnic groups display a resistance to P-noliside and P-cynomolgy in addition to P-vivax. Next slide. So the final evidence came from Lou Miller groups, which show first by using P-noliside, which we are able to cultivate, that this parasite was not able to infect erythrocyte from Duffy negative African, while this parasite easily infects erythrocyte from Duffy positive. And the final evidence comes from a clinical trial. Lou Miller infects 17 volunteers with different Duffy blood group phenotype. And what is shown is that the 5 Duffy negative African Americans do not develop a P-vivax infection, while the 12 Duffy positive individuals develop a blood stage infection. Next slide. So the next step was to understand, and in the 90s, the parasite ligand interacting with the Duffy blood group was described. First for P-nolesi, and there is 3 different ligands, the PK, DBP alpha, beta, and gamma, and for vivax, the Duffy binding protein. And what has been shown that it's the PVDBP is expressed on the surface on the merozoite. Next slide. So at the end, we were able to understand the invasion pathway of vivax, which is mediated by the interaction between the Duffy binding protein and the Duffy antigen. So this was the dogma. Next slide. Next slide. So recently, recently we understand why the vivax show a strict tropism to reticulocyte, and this tropism is mediated by the interaction between the PVRBP2B, which bind the transferrin receptor. And this evidence was published recently in Science. Next slide. So the dogma was that Duffy negative are naturally resistant to vivax invasion, and all the vaccine strategy was based on to block the interaction between the PVDBP and the Duffy antigen, and also the PVRBP2B, with the transferrin receptor. But next slide. With the emergence of the molecular diagnostic method, we have a new idea of the landscape of and the epidemiology of P-vivax, especially in Africa. And over the past 20 years, there is growing evidence that P-vivax is circulating in Africa and in Duffy negative people. Next slide. So I used an excellent review from Twig, which compare all the reports of P-vivax infection in Africa. So this report from local P-vivax clinical case report, for community survey, or for P-vivax infected vector, or by serology. Next slide. And they show that, so they detect P-vivax cases in 17 countries in Africa. So you have the list here. In 13 countries by survey, and in vector and serology. So that means that P-vivax transmission is ongoing in Africa. And what they observe is that P-vivax in Africa is widespread, diverse, and likely often undetected because the people have a very low parasitemia. Next slide. So the dogma is not relevant now, because the Duffy negative phenotype is not a definitive barrier to Plasmodium infection. So that what we know is that the absence of the Duffy antigens appear to render the invasion mechanism less efficient, but not null. And the question is that is historically not detected, or it's a recent adaptation of a few strains able to infect Duffy negative red blood cell. So now we have to understand what is the pathway used by the parasite to invade the Duffy negative reticulocyte. And on the right, you have the list where P-vivax infection was described were described in Duffy negative. Next slide. So the hypothesis is to link to the invasion of the Duffy negative. There is some observation in the 90s showing that when we did a KO of PK alpha Duffy binding protein, the parasite is able to invade rhesus erythrocyte, which means that the parasite use alternative pathway. And the same thing is that with the Cymeri erythrocyte, which show that there is no binding of the PVDBP with Cymeri erythrocyte, which can suggest that the Cymeri may model the Duffy independent pathway. Next slide. So the hypothesis that groups are exploring could be based on two ideas. The first one could be that we have a Duffy-dependent pathway, which means that Duffy-negative phenotype do not mean that there is no antigen on the surface of the reticulocyte, but there is a weak expression of the Duffy antigen. And what we observe is that in the PVDP gene, the amplification of the DPB gene, which means that this amplification means an increase of the amount of the DPP protein on the merozoite surface, which can be a balance to invade the Duffy negative reticulocyte. So we have observed multi-copy of PVDPP in several countries, in Madagascar and Ethiopia, where we can find Duffy negative people with Duffy positive people, but we also find amplification of this gene in countries where only Duffy positive people are living. So it doesn't seem that it's a good hypothesis. Next slide. The alternative hypothesis is a Duffy independent pathway. So there is different hypotheses. The first one could be that we have a specific polymorphisms in the DBP region 2, which could promote the binding of the parasite, the merozoite, on an unknown reticulocyte receptor. Next slide. Hypothesis 2 is linked with the discovery of a new erythrocyte binding protein, which named EBP or DBP2, which have the hallmark of a ligand able to be involved in the reticulocyte invasion. And this ligand could bind an unknown receptor on a reticulocyte. What has been shown is that the EBP is able to bind Duffy positive and Duffy negative erythrocyte, but at very low level for Duffy negative. And another group showed that EBP bind at high level immature reticulocyte, but only for Duffy positive reticulocyte. That means that it's not validated, and we have to confirm this data. Next slide. Hypothesis. Next slide. 3, yes. No, previous one. Some two ligand has been described to bind both Duffy positive and Duffy negative, which is the PVMSP1P and the PV gamma, but we have no confirmation with functional study. And the next slide, the last hypothesis could be that an unknown P-vivax ligand binds to an unknown receptor of reticulocyte. Next slide. So the problem to date is that the infection, the invasion of vivax, of Duffy negative red blood cell by Plasmodium vivax is an enigma. And the main challenge that we have no validation by functional study, which is linked that we have no system to produce a long-term P-vivax in vitro culture. So several groups are working on this issue, and the alternative option could be to use next generation like genomic and transcriptomic study. Genomic could be used to characterize lossy under selection, and transcriptomic study could be used to define the gene expression profile of P-vivax isolates collect from Duffy positive and Duffy negative. And the other alternative is to use animal models like primate model or humanized mice. Thank you.
Thank you very much, Didier. We can go ahead with the next speaker, Eskil Pedersen. Eskil is connected. Yes.
Okay, can you hear me?
Yeah, now, yes. Okay, Eskil Pedersen is from Institute of Clinical Medicine, Faculty of Science, Aarhus University in Denmark, and also visiting professor at the Department of Molecular Medicine, University of Pavia in Italy. This talk is about the risk of Plasmodium vivax reemergence in temperate climate areas. Please, Eskil.
Thank you very much. Thank you very much for inviting me to this interesting symposia. Can I have the first slide? This is just to show that vivax has been for several hundred years in Europe, and this is a painting by Dürer showing his painful spleen. Next one, please. Next slide, please. This is a map of the distribution of malaria in the pre-control times, and you will see that you have in China, in Europe, and in North America, there was huge areas that was endemic for malaria. Next one. This is maps from a study last year comparing the distribution of vivax in 2005 and 2017. And you will see that it has mostly disappeared from the temperate areas. So what is the risk factors? What is the risk that we get it reintroduced? Next one. This is a paper by Nick White, pointing out that we have two different forms of Plasmodium vivax. We have a tropical form which has a short incubation period, and we have the long latency Plasmodium vivax, which incubate in the liver for maybe 6 to 8 months, allowing it to incubate over the winter and give an outbreak next spring where we have the hot weather. So it's important to realize that we have these two forms, and in some places, like in India, we have both forms. Next one. To the left, you will see the different phenotypes of Plasmodium vivax. This is a paper by Bray from 1982, and they also call the long latency temperate forms Plasmodium vivax hibernans because it's hibernating over the winter. And on the right hand, you see a graph of Plasmodium vivax relapses in the U.S. servicemen returning to the U.S. from the Korean War. and you will see that they were going back to the US in 1950, but they had the relapse in 1951. So they were all infected with long latency temperate form of Plasmodium vivax in Korea. Next slide. And you can actually distinguish these two forms by using haplotyping, and of course that's a strong tool. So I think it's very important when you have an outbreak of P. vivax, you have to do genotyping, because if you have a long latency P. vivax, you have to look for the introduction, but maybe 6 to 8 months previously. If you have a tropical P. vivax, the incubation is much shorter. So this is an important tool to find out when did the introduction happen. Next one. We have had a Plasmodium infection in the Netherlands, and there was an outbreak during the Second World War. But you can see that there's been an outbreak since in the past century several times in the Netherlands. But in 1951 it was all over, and we haven't seen local malaria in the Netherlands since then. Next one. Next. You would not think that Finland was a place. Previous one, please. Previous one. Previous slides. Thank you. You would not think that Finland was a vivax and malaria endemic area, but on the upper left you can see malaria cases in the Finland map for about 150 years. And on the right side, you have the malaria cases during the Second World War. Finland was engaged in a intensive war with the Soviet Union from 1939 up to 1948. And in 1940, the incidence of malaria in Russia was 20 per 1,000 inhabitants. So a lot of the Russian soldiers sent to the front was infected with malaria, Plasmodium vivax, and the mosquitoes bite the Russian soldiers and introduce the vivax on the Finnish side of it. But you also when you had the indigenous autochthonous Plasmodium vivax in Finland, they actually showed that the mosquitoes could survive in the houses where it was hot enough that you could have a sustained transmission even during wintertime in the houses. Next slide, please. Next slide. This is a paper from 1949 showing the autochthonous and introduced malaria in Germany in 1946-47, and this is from the Berlin. Greater Berlin, Mecklenburg, and the reason was returning troops from Russia, from Southern Europe, who was malaria positives. And you can see that they started secondary transmission, and they estimated in 1946 that 54% was autochthonous, and in 47 that 56% was autochthonous. So this is an example of how you introduced malaria by a large number of people being infected, returning to an area where you have a lot of susceptible people. It's noteworthy that these outbreaks of malaria in Germany was all over in 1950. And you can speculate why was it possible in the conditions just after the Second World War to control malaria in Germany. And my thought is that all these cases, they were symptomatic. We had no people with immunity, so they were easily recognized and treated with quinine. Next one. Again, you send a lot of non-immune people into endemic areas, and this is number of malaria cases in Allied soldiers in Italy, and you can see it peaked in 1943, where you have more than 30,000 cases of malaria in Allied soldiers. Of course, they were infected in Sicily and so on, Italy, despite prophylaxis, but then they got it under control by increasing the discipline, and that helps. So that's another way of looking at it, that if you send non-immunes into an endemic area, you can expect infections, of course. Next one. And this is another example from the 1990s. This is an outbreak in Azerbaijan. There was in the course of the breakup of the Soviet Union, there was a war between Armenia and Azerbaijan, and that caused an increase in the malaria cases, which is due to breakdown of control measures. So there was an outbreak here with more than 30,000 cases. Next one. This is the outbreaks, and on the left hand, the next speaker is going to speak about the outbreak in Greece in 2011-12. And on the right hand, you can see that in the ProMed, there's some isolated reports about imported or autochthonous cases, which were diagnosed but never really resulted in a large outbreak. So we see autochthonous cases here and there, probably secondary cases from imported cases, but it has never really caused a major outbreak, because in our modern times, where we have access to anti-malarial drugs and easily diagnosis, and people in Europe are not immune, so all cases will be symptomatic. Yes, next one. I was involved with Stéphane Picot in an outbreak in Oman in 2015, where there was more than 50 cases of Plasmodium vivax, and Stéphane did the genetics, and you can see here that we could type this, so showing clearly that it was a short, the tropical format Plasmodium vivax. It did not cluster with a long temperate Plasmodium vivax found in North Korea. So it was a tropical form, and it clustered closely to sequences from Iran. Not that we could prove that it was somebody from Iran working on this construction site, but it just shows you that the molecular typing of the parasites will be an important tool when you have an outbreak, and especially to distinguish between the temperate and the tropical form of P. vivax. Next. So where do we have the biggest risk? This is P. vivax in the demilitarized zone in South Korea. And even though this is some years ago, it's quite clear that you have temperate form of Plasmodium vivax in North Korea, and these cases here in South Korea is because infected mosquitoes have been crossing the border and giving rise to cases in South Korea. So there's no doubt that North Korea these days are probably the main focus of the temperate form of the Plasmodium vivax. Next slide, please. And this is in a model based on where we could expect to have vivax if you have a large number of refugees from North Korea spreading into China. So this is how you will see a spread of Plasmodium vivax. It's movement of people. The mosquitoes are there, but the movement of people is what determines whether you will have a reintroduction or not. Thank you very much.
Okay, thank you very much, Skilled. So we can go ahead with the next presentation. is the Dr. Danai Pervanidou from Vector-Borne Diseases Department, the Directorate for the Epidemiological Surveillance and Intervention for Infectious Diseases. This is the Hellenic National Public Health Organization in Athens, in Greece. So the presentation is about Plasmodium vivax reintroduction risk in Greece. Just a few seconds. I would like to remind to the people who are connected via streaming that you can send your question by the email highlighted in this picture. So please send email via this. Send your question via this email. Thank you. Thank you, Danai.
Hello, everyone. So can you hear me?
Yes.
Okay. First of all, thank you for the honor to speak and participate to this meeting and share the experience of Greece regarding the recent recording of reintroduction events of Plasmodium vivax and how we manage this risk. Next, please. A quick overview. Greece is malaria-free since 1974. Since then, several imported cases are reported annually to the Hellenic National Public Health Organization, referring to patients infected abroad, either migrants from endemic countries or travelers. In addition, since 2009, events of Plasmodium vivax reintroduction have been recorded in rural areas in the country, concerning mainly sporadic cases, but also small clusters. So what happened? And Greece, a European malaria-free for decades country, had to face this risk. Next, please. So what happened was the importation of Plasmodium vivax in receptive areas. During the last 2 decades, a large number of migrants entered Greece, and especially since 2006, there was an increased migrants influx from the Indian subcontinent, from Plasmodium vivax endemic countries of the Indian subcontinent. Mainly undocumented migrants work as seasonal land workers in rural receptive areas of the country. In accordance with this increase of the migrants' influx, since 2009, more than 75% of imported cases were actually among migrants from endemic countries, and 77 of them originating from the Indian subcontinent. Next, please. You can see here the numbers of imported malaria cases and Plasmodium vivax malaria cases in Greece since 2009. 62% of all the imported cases were actually Plasmodium vivax cases with the blue bars. And as you can see in the table, there was an increase in their number in 2015, 16, and 17. And this was due, next please, to the recent, next please. Next slide. This was due to the recent refugee migrant crisis since 2015, when around 1 million sea arrivals were recorded in Greece during 2015, 16, 17, and 18, with 1/3 of them originating from malaria endemic countries. Most of these refugees and migrants actually were relocated in other countries, but according to recent estimates, more than 100,000 refugees and migrants that arrived in Greece since 2015 still remain in Greece, both in organized accommodation facilities and camps, but also almost half of them outside of organized accommodation facilities. Next, please. So, as I already mentioned, in addition to these imported cases recorded, a number of locally acquired cases have been recorded in the country since 2009. And the vast majority of these cases, actually all but one, were Plasmodium vivax cases. Next, please. As you can see, the numbers of the locally acquired Plasmodium vivax cases in Greece peaked in 2011 and 12, and then their number declined steadily following the implementation of intense public health measures. Next, please. The vast majority of, next, please. Next slide. The vast majority of locally acquired, next slide. Okay, the vast majority of locally acquired Plasmodium vivax cases recorded in only one municipality, in Evrotas municipality, southern Peloponnese, in 2011 and 12, where the 74% of all Plasmodium vivax cases were recorded. Actually, 36 cases in 2011 and 10 locally acquired cases in 2012, along with cases recorded among patients from endemic countries considered at that time as imported cases. All of these cases were recorded in 5-6 small villages within a 2 to 4 kilometers distance in the area. At this point, I would like to clarify that regarding the case definition of imported cases, we use, of course, the WHO case definition regarding patients infected abroad to malaria endemic countries, but also we use epidemiological criteria to further define as imported Plasmodium vivax cases among migrants occurring in migrants with onset of symptoms within 3 years after their arrival to Greece, so to include also the relapses of the Plasmodium vivax. Next, please. So, next slide. So let's zoom into Evrotas area to understand the risk factors that contributed to this local transmission. To begin with, Evrotas was a historical hotspot of malaria before the elimination of the disease in Greece. Actually, the affected areas is a plain agricultural rural area of 20 square kilometers near the delta of Evrotas River, with a large network of irrigation and drainage channels serving as Anopheles mosquitoes breeding sites, and especially Anopheles zakharovi, that is considered the main vector, convenient vector of malaria in Greece. In addition, in Evrotas, there is a large number of migrants from endemic countries, from the Indian subcontinent, and mostly from Pakistan, who live there and work there as seasonal agricultural workers during summer months, from late spring to mid of autumn months. And actually, most of them are undocumented migrants living in not so optimal conditions in some occasions, and in close proximity to Anopheles breeding sites in the area, and directly exposed to their bites. Next, please. Next slide. So actually what happened in Evrotas and was revealed by genotyping was that in 2011 and 12, an event of indigenous transmission occurred of 2 haplotypes as indicated by genotyping results, as some of these locally acquired cases belonged to 2 family clusters, but also in these clusters belonged not only Greek residents, but also migrants from malaria endemic countries, actually indicating that these migrants, some of these migrants were not actually locally acquired, were not actually imported, but were locally acquired, so they were reclassified. Also, genotyping revealed that these isolates passed from one year to the other, from 2011 to 2012, posing a risk for establishment of these strains in the area. Next slide, please. The establishment was prevented, the transmission was interrupted after a series of intense and resource-demanding public health interventions that are also continuously implemented in the area, actually. And as you can see, since 2013, only one Plasmodium vivax introduced case was recorded in 2015, and it had a different haplotype from the previous ones, and only imported cases were recorded in the area thereafter, or all with different haplotypes. Next, please. Next slide. So beyond Evrotas, in other areas where locally acquired cases have been recorded since 2009 in Greece, All cases were introduced cases, actually, the first generation of transmission. All areas were rural areas, agricultural areas with a combination of the risk factors, so Anopheles breeding sites near to wetlands, and also migrants population from malaria endemic country. What is important and interesting is that most of these areas are areas that were also historical hotspots of malaria before the elimination of the disease in Greece, as you can see in the red areas in the map, indicating that these areas has a high malariogenic potential in case that Plasmodium vivax patients enters in the area. Next, please. Next. Next slide. Okay, here is another example of introduced cases. A cluster of 8 introduced cases occurred within 3 weeks in a Roma community in North Greece in October 2018. All of these cases were located in the bottom right corner of the Roma community and in close proximity with a migrant detention center, as you can see with the yellow pin. And the hypothesis was, again, that Plasmodium vivax was imported in this detention center and then further transmitted in the Roma community, as also the Anopheles presence is there. Next, please. Okay, so an overall diagram with the cases, the Plasmodium vivax cases in Greece, imported and locally acquired. The peak of the cases, of locally acquired cases, was in 2011 and 2012. After this peak, cases declined steadily over the next years, following the implementation of intense public health measures, and the number of introduced cases remained low, despite the migration crisis and the increased number of imported cases in 2015, 16, and 17, indicating that the well-established measures actually may contribute actually efficiently to the prevention of further transmission of Plasmodium vivax. Next, please. Next slide. Next one. The next one. Next slide. No, the other way. These are the previous slides. Next slide, please. And the next one. Next one. Okay. So following the occurrence of these cases, of these local acquired cases in 2011, a national action plan for the management of malaria was developed and is continuously implemented from the Ministry of Health and the National Public Health Organization, including a series of activities. Next one, with the collaboration of many stakeholders at national, regional, and local level. Next one, please. So all areas in Greece are aligned to risk level, which is having into consideration the vulnerability and receptivity of the area and also the recorded cases. And this risk level also defines the activities to be implemented. Next one. Enhanced malaria surveillance is implemented in Greece since 2011 in order to timely detect and diagnose the malaria cases. We perform this by raising awareness of health professionals to test all suspected cases, and also with active case detection activities, which include also the reactive case detection after the notification of each locally acquired, of each introduced case, a fever screening in the neighborhood, and also proactive case detection in high-risk areas such as Nevrotas area, where fever screening is performed on a regular basis. Also, we try to screen the newly arrived migrants from malaria endemic countries arriving at the Greek-Turkish land borders and sea borders in 2012 and 13 through rapid diagnostic tests. But unfortunately, among the over than 3,000 migrants that were screened with rapid tests, no one saw the positive result, indicating that actually we don't have a valid tool to diagnose the asymptomatic carriers. So we interrupt actually this activity. We investigate every case and we perform case-based risk assessment for local transmission. Next, please. Next slide. Okay, here you can see the next one. This one. Thank you. Here is the exhaustive reactive case detection implemented in the Roma community. Actually, during the reactive case detection, we identified 2 of the 8 of these introduced cases, and then we also continue with this activity in the following year. Next one. Next slide. In Evroras area, since 2011, a proactive case detection activity is continuously implemented with the involvement of many agencies. This activity is actually it was founded by the National Public Health Organization at the beginning, and now by the Peloponnese region. And actually, there is a field team from April to November and performs fever screening every 7 to 15 days to all migrants residing in the area, and the field team is also on call 24/7. And all symptomatic cases are tested with rapid tests and then with microscopy, and also the confirmed cases are treated by directly observed treatment, as most of them do not require hospitalization. Why we had to go to the migrant community and implement such a resource-demanding activity? We saw that in 2011, there was a diagnosis delay between the symptom onset and diagnosis up to 6 days. Median time was 6 days. And this was because of the limited access of migrants in health services due to their fear of arrest, their fear of cost of the hospitalization, language barriers, and transportation barriers. Next, please. So we implemented this measure, and actually it was not an easy activity to perform, as this population is highly mobile. But still, we have significant results, and the median time from symptom onset to diagnosis actually decreased from 6 days to 3 days in 2012, reaching the WHO target, and to half a day actually in 2017. Reducing dramatically the transmission risk and the infectiousness period of the migrants. Actually, almost 90% of cases among migrants in the area were actively detected through these activities. Next one. This is an overview of the public health intervention in Evroras. As you can see, in 2011 and 12, there was the peak of the locally acquired cases and the event of the indigenous transmission. Intense mosquito control was implemented in the area, and as well as active case detection. But these classic standard malaria prevention measures did not manage by themselves to bring the cases to zero. As you can see, in the next year, in 2012, the number of local acquired cases was decreased, but still 10 cases were recorded, posing a threat, posing a risk of establishment of Plasmodium vivax in the area. So in the next year, these classic control and prevention measures were also enforced by the deployment of mass drug administration among migrants, targeting migrants from malaria endemic countries residing in the area, after, of course, their consent. And this activity, this combination of activities also was performed in 2014 and 15. As you can see, since 2015, no further cases were recorded, local acquired cases, introduced cases were recorded in the area, indicating that this combination of interventions were actually successful and effectively contributed to the prevention of local transmission in this area. Next one. Next one. Very important is also to have enhanced laboratory diagnosis capacity. We supported the malaria reference laboratory for free of charge diagnosis for every patient, every suspected case. And you saw the significant information of the Plasmodium genotyping also. Also, to enhance the local diagnosis capacity is crucial, and we further train microbiologists on blood smear and also distributed rapid diagnostic tests to over 200 health units and migrant camps. Next one. Next one. Case management. We provide free access to diagnosis and treatment for all patients, and the treatment is standardized, including both chloroquine and primaquine, of course. Next one, please. Next one, communication activities for the locals and for migrants. Next one. Blood safety measures are implemented around local acquired cases for some months. Next one. And vector surveillance and control activities, next one please, are implemented and intensified by regional and local authorities. And also, in addition, in Evroras, indoor residual spraying is performed in the migrant shelters, as well as distribution of long-lasting insecticide-treated nets. Next one, please. So also in areas with refugee migrant camps, intensified activities are implemented since 2015. Enhanced surveillance, distribution of rapid tests, distribution of mosquito repellents, and intensified vector control around the migrants' camps. Next one. Actually, no local acquired case was connected with organized accommodation facilities. To conclude, areas at risk for introduction exist in Greece, where the competitive vector is combined with presence of malaria patients coming from endemic countries. Sporadic events of Plasmodium vivax reintroduction have been recorded also in Greece. One event of indigenous transmission was recorded in one area in 2011 and 12. And after the peak of cases in 2011 and 12, the number declined steadily. However, a few introduced Plasmodium vivax cases have been also recorded during the last years, indicating the need to sustain the prevention activities. Next one. Good practices in Greece include action plans, established procedures, multi-sectoral collaboration, regular and constant awareness raising of health professionals on the malaria diagnosis, and free access to migrants to health services. Challenges include case finding, as asymptomatic and mild Plasmodium vivax relapses are not easy to be diagnosed, and no screening tool exists for carriers. Migrants' awareness that they have access in our health system, and communicating the risk to the public and to authorities. Next one. Globally, we think that Greece example is a fine example that also confirms the fact that Plasmodium vivax has actually a high potential to initiate and maintain local transmission, to establish sustained transmission within a short time, in vulnerable and receptive areas, in areas with high malariogenic potential, when carriers of Plasmodium vivax enters these receptive areas. Asymptomatic or mild relapses of Plasmodium vivax result in extended periods of infection and transmission. So in Greece, as well as in other countries, also in other European countries where recent recordings of introduced cases of malaria were also recorded, and in other malaria-free countries, continuous vigilance is crucial, especially in vulnerable and receptive areas, in order to prevent the further transmission of Plasmodium vivax when it's imported and introduced in the area. Multi-sectoral strategy is needed, a combination of many pillars of response to early detect the cases, properly treat the cases, investigate, case-based investigate the cases and assess the risk, and also implement an integrated vector control. Next one. Thank you. Our surveillance reports are published in our website, also in English, and you can see some of our informative leaflets here. Thank you very much.
So thank you very much, Danai, to present us what could happen if the Plasmodium vivax could reemerge in some areas. There's definitely a lot of work to control to carry out surveillance activities. We can go to the last speaker, the Dr. Ahmad Raisi from National Program Manager for Malaria Control, the Ministry of Health in Tehran. So his talk is about the Plasmodium vivax malaria in Iran. Please, Ahmad.
Thank you so much. Thank you, Carlo, for inviting me. That's my pleasure to be with you in this very important vivax meeting. Are you hearing me?
Yes.
So, and this is for me is also the good memory from Istituto Superiore di Sanità that, as you may remember, 29 years back, we were together in the same class of basic course of malaria that was run by WHO. So that is also the good opportunity to remember the memory of Giancarlo, that he was tried too much for that course. So I'm going to raise some summary information about malaria and history of vivax malaria in Iran. Malaria has been a widespread epidemic disease in Iran for at least 3,000 years. And we can see the several notes in the history of malaria. For example, in the Avicenna book, it's written about malaria. Of course, that was before exploring malaria parasite. And if you go to the historical document, for example, you can see that about 20,000 people who had been relocated from Armenia, they died from malaria in the Caspian place in the late 18th century. For sure, by the early 20th century, the disease was the most important public health problem. At that time in Iran, 30% of population got malaria annually. It means that incidence of malaria was more or less 5 million cases. The population was 15 million, and annually 5 million people got malaria. So, with this history, I want to show you the next slide, please. I want to show you that with this, we can see that how we can reach to some level of successful efforts. As you see the second slides, from 2018, we did not have any report of single indigenous case of malaria. And if we can keep this situation this year, I mean in 2020, for another 2 months that we are waiting to finish it successfully, then we will be eligible to apply for malaria certification. malaria-free certification. So that's the huge successful efforts. We started from 5 million cases per year, and now for 3 consequent years, we did not have even a single indigenous cases. Please, next slide. You can see here that the majority of malaria cases in Iran are vivax malaria. Unfortunately, issue of vivax malaria still is neglected. And I'm so happy that Carlo and our colleagues raised this issue, this important issue, to re-emphasize on importance of vivax malaria. That's very important. Everybody in the Western country, especially in US, when they are working on malaria, they are focused on falciparum malaria. For sure, falciparum malaria is a killer disease, and we have to pay attention more. But vivax malaria is another type of malaria which is difficult to control it, especially in temperate area. Next slide, please. The majority of cases during these recent years, as showing from 2015 to 2020, we can see the majority of imported cases, especially recent 3 years. All cases of malaria are imported. But this importation does not mean another nationalities. You can see here, for example, in 2020, around 510 cases till November are imported cases, but regarding nationality, they are Iranian. So it seems that importation is different scenario to nationality. The majority of these people that they are Iranian and got malaria in the other side of the borders are the people that they are involved in the illegal fuel transportation and trading. Next, please. 3 days back, I was visiting the border area between Iranian Baluchistan and Pakistani Baluchistan. And I have seen that, as you can see in this slide, the all available measures for malaria elimination is doing there by my colleagues. For example, in case of surveillance and case detection, there are proactive, passive, reactive, intensified case detection, and everything. The single case by case, has to be investigated and send the investigation form as the classification form to the central level, to the provincial, and then to the central level. So it seems that we have missed something, and I'm thinking that in this way, my 30 years experience is not enough to combat with the vivax malaria, which is focused in the southeastern part of the country. Why? Because it's related to the special population, fuel smuggler, seasonal, unregistered foreign workers. As some of our colleagues mentioned, a lot of Pakistani unregistered foreign workers are engaged in the bananas farm, and they are working as the illegal workers, but they are cheap. So it means that in this case and in this situation, we need to have some new tools. And I'm thinking that maybe current tools, current interventions are not efficient to combat this last foci of vivax in the country. Next slide, please. As you see here, for example, we are using RDT. Of course, the main and the gold standard still is microscopy. But RDT is available for all communities, for all people and fuel smuggler group. And we train them. We distribute RDT among themselves to check their situation. And in case of having positive RDT result, then they call the first available health staff to come to treat them. It is one of the innovations that we started to have it. And for sure, blinded cross-checking of the all slides that are coming from Pakistan side is the other thing that our quality control system is doing. The other innovation that we have to start it, and we have already started for this important area of last vivax malaria foci, is the targeted presumptive treatment with primaquine that is administered by staff, and it is under direct supervision for having efficient as and enough precaution regarding G6PD deficiency. The next slide, please. Here you can see that this vivax malaria is limited in the some districts close to the border of Pakistan. And you can see here that the all imported cases have direct link to population movement. It means that our colleagues check the all importation with the local situation, foci investigation, to prove that this importation has any link to population movement or not. And you can see here in the map that our colleagues have already investigated the all local foci to prove and to show the linkage between this importation and reported cases. Next, please. Of course, the challenges and bottlenecks in elimination and prevention of re-establishment of malaria, which is focused on mainly on vivax malaria, is the first one is population movement and cross-border mobility. I have to say that it's not possible to keep the border. It's not possible to build a wall in the border. They are the same community, they are the same culture, they belong to the same religious, and they have to go and come back. And unfortunately, in this situation, the other type of population movement, like fuel smuggling, is very efficient to suppress our efforts toward malaria elimination. One of the most important things that we raised here today in the meeting by some of our speakers is the role of molecular marker. And really we need to focus on molecular marker to confirm that is there any linkage between the cases that we define them as introduced cases or the cases that they are result of first generation of introduction or transmission. So this deficiency is one of the most important points that we need to have more focus scientifically on this molecular marker to show and to approve that the case are introduced, not indigenous. And for sure it is not so easy. And we have previously discussed with some of our colleagues that we have to set up a system to do this molecular marker. For sure, we have the ability to confirm the type of case genetically. For example, is the mixed infection, vivax or falciparum alone, for example. But we need to do more focus on molecular marker to prove, to confirm that there is not result of local transmission. Next slide, please. Immigration from endemic neighboring countries by unregistered laborers and drivers engaged in border trade are very important, and it is there is a lot of risk of malaria reestablishment even in the cleared up receptive foci. Need for effective collaboration with neighboring countries to share knowledge, skills, and in context of case detection, management, case management, early warning system, genomic initiatives, and vector control interventions is essential. We set up a network, a local or regional network between Afghanistan, Iran, and Pakistan. We call it PIDAM net. We have sorted a lot of meetings, but the only results that I can say is training of some of experts, some of the managers in those two countries that we invited them to sit on the international course that we normally manage it with collaboration of WHO, and to certify them as the graduated manager experts from the course of malaria case management, malaria vector control, malaria diagnostic, and a long 2 months, it's 2 months, and a longer course of 2 months of international course on malaria management and control. Some efforts were initiated by WHO, UNDP, GFATM, Global Fund to operate regional coordinating in recent years. But as I told you, it has never resulted to an effective action we have some initiative like PMNet, G5 forum, but it was not effective as we were waiting. Thank you so much indeed, and please accept my apology as I was more focused on managerial control and elimination aspect.
Okay, thank you very much, Ahmad. Thank you. So we have very few minutes for some question. I invite all the speakers to turn on the webcam. In this way we can together. Okay. So we have a couple of questions from the streaming to Dr. Nunu. So the question is, how long after the first PVIVA infection relapses can occur?
2 to 3 weeks, they say, as the earliest you can detect it with confidence that it's a relapse, provided you treat the primary infection with a fast-acting drug. Mm-hmm. So the relapse can occur very early on. This has been seen with the Chesson strain. And I have to say, I'm a bit uncomfortable with defining all the strains as either tropical or temperate. The Madagascar one, which might well not be from Madagascar, is kind of in between. And I suspect there are in-betweens and gradations between temperate as the hibernans and the Chesson strain. I suspect that there's every single combination you can imagine. But 3 weeks, I would say, is the earliest you can have as a relapse, what you can call a relapse.
Okay.
You
can direct address your question to the.
Yes, it's for George, because the relapse, maybe it's because people are treated by chloroquine, so maybe it's linked to the half-life of the chloroquine. No?
That's why I said if it's a fast-acting drug that's treat the primary infection, like artesunate, then you can be sure that it's vivax relapse afterwards. If the primary,
you have an idea that maybe by treating by artesunate that we have a shorter time for the relapse. You have some evidence about that?
No, because the studies, not to my knowledge, the studies haven't been done where people have been infected on purpose with Plasmodium vivax, treated immediately as the parasites appeared, and then left to see what happens. Of course, the hepatic schizons don't all grow at the same rate. So if you have some that take 15 days to grow, then you relapse, or the primary infection will be 3 weeks later. This is also another misconception, or potential misconception, is that for falciparum by day 6, all the hepatocytes, infected hepatocytes, have matured. In vivax, that might be take 3 weeks, 2 to 3 weeks.
Okay.
Difficult to study.
You have also a question for George. I mean, I received the question that does P-vivax located in the spleen affected by anti-malaria treatment or protected?
If one believes fully the Ascoli treatment, then it's not fully affected or as well affected. It's difficult to say because we as I say, the demonstration of parasites in the spleen, as a high biomass of parasite in the spleen, is very, very recent. This is done with work carried out by Stephen Core and colleagues, Mike Anstey and Nicholas Anstey and Rick Price in Papua in Indonesia, where they found that there's maybe 1,000-fold more parasites in the spleen than there are in the circulation on the day that the spleen was removed. This was done by splenectomy. The splenectomy was done for other reasons. So therefore, this is early days to say whether what's in the spleen is resistant to the drug. I suspect that they're not that resistant, but they might well be more protected. Or the other thing is that if they survive, they might remain in the spleen and not go out to the circulation. These are very early days and an exciting area of research.
Okay. Okay. We have a question from Didier Menard. You know what is the percentage of Duffy negative people positive for vivax in observed in your study?
So I have the figure from Madagascar when I did a study in 2010, and we found more than 8% of Duffy negative were positive for vivax. It was a clinical cases, but I have no idea in the other country.
But did you know if they are located in some specific areas or was scattered around?
It seems that we can see clinical cases in area where you have a mixed population of Duffy positive and Duffy negative, and it's what we saw in Madagascar. In area where you have 75% of Duffy negative and 25% of Duffy positive, and it's the same thing in Ethiopia.
Okay.
Because there is a lot of report from Ethiopia.
Okay. And I think that's the last question to Danai. Did you evaluate the origin of vivax strains at the time delay for relapses?
I didn't hear the question. If we identify the origin?
Yeah. The strains identified in your country.
I didn't understand the question. One more.
So the question is, did you evaluate the origin of the vivax strains responsible probably for the outbreak of malaria?
Yes, actually, as I saw, we performed some genotyping of many samples of Plasmodium vivax in both the locally acquired and imported cases in the country, and it was shown that many important information came from this genotyping. If this is the question that I understand regarding the connection of cases, and that some cases were actually clusters and multiple introductions occurred actually. But regarding, if they mean regarding the origin, if they were from a country, from which country, I'm not. I don't know. No, I don't think that we did that.
Yeah, okay.
I don't know the patients. The vast majority of the imported cases were actually from Pakistan, but regarding the genotyping results on the origin of the strains.
Yeah. Okay. The very last question. You know what was the vector density in these areas? I mean, you observe high density of vectors was Anopheles sakharovi.
Actually, at the first years, 2011 and 12, in the Evroras area, the population of Anopheles was not high that we could catch on the traps. We find out that transmission can also occur either in areas with medium, let's say, population, but a high importation risk, a high number of cases, of imported cases, or in areas with a low importation risk, 1 or 2 imported cases, and a high Anopheles population. So we had areas with both the combination of these risk factors.
Okay. Okay, thank you. Sorry, I think we have to stop the. Okay. Thank you very much for all the speakers. I think that thanks to all this presentation showing the diverse expertise, I think several key features of the vivax biology and epidemiology has been discussed during this webinar that focused on Plasmodium vivax, and I hope that this has been interesting and informative enough. So thank you very much. Bye-bye. I hope the next time that will be in presence. Bye-bye.
Thank you, Carlo.
Thank you very much.
Thank you, Carlo.
Bye-bye.
We hope so too.
Bye.
Bye-bye. Bye-bye to everyone.